So in this lecture segment, we're going to look at the electron transport chain, chemiosmosis, and oxidative phosphorylation. That is what I have as part 4, the last part of cellular respiration. As you can tell from the name, it's kind of complicated. Let's start with the electron transport chain, or ETC. In the inner membrane of the mitochondrion, you have a lot of transmembrane proteins and integral proteins that are electron carriers. Ultimately, what's happening in the electron transport chain is the electrons that are being carried by NADH and FADH2 are released by those carrier molecules and transferred to the proteins of the electron transport chain. And we usually call those proteins of the electron transport chain complex 1, complex 2, complex 3, and complex 4. So there are 4 of them. So the NADH and FADH2 drop off their electrons either at complex 1 or complex 2. And complexes 1, 3, and 4 are proton pumps. And you'll remember that a pump moves something against its gradient. And in this case, protons, hydrogen ions, are being moved against their gradient. And it maintains a gradient of protons across the inner membrane. You have a lot more protons in the intermembrane space and less protons in the matrix. And these complexes are pumps to keep that gradient in place. So here is kind of a diagram of the electron transport chain. You can see the inner membrane is shown here, the phospholipid bilayer, and you can see the way they have this oriented. The intermembrane space is up above and the matrix is below. Of course, you could flip this and look at it the other direction, but that's the orientation of this image. So the intermembrane space has a lot more hydrogen ions. So you want to imagine the gradient, a lot of hydrogen ions up here and not as many in the matrix. And so in pumping hydrogens into the intermembrane space, that means that these protein complexes 1, 2, 3, and 4 have to, actually not 2 doesn't pump anything, 3 and 4, they have to use energy to move protons against the gradient. So that's active transport. So you have this pumping that's going on. At the same time, you have those electrons that I mentioned. The electrons are being moved from complex 1 and 2 to complex 3 and complex 4. So the protons are moving along the membrane. They're kind of bouncing from one protein to the next. The protons are moving across the membrane. So electrons moving along the membrane, protons moving across the membrane. So 2 things happening at the same time. The reason the electrons move along the membrane is that as they move from one protein complex to the next, some of their energy is taken by the pump and used to do that pumping of protons across the membrane. So the energy from the electrons is sort of stolen from them and used to pump protons across the membrane. By the time the electrons get to complex 4, they have virtually lost all of their energy. They've given it away, in a sense. And so at the end of complex 4, those electrons are low energy electrons. When they come in on NADH, they are high energy electrons. When they get to complex 4, they are low energy electrons, because their energy has been used to pump protons across the membrane. And so at that point, those 2 electrons are combined with 2 protons and some oxygen to make water. So water is a waste product, a way for the cell to get rid of those low energy electrons. But ultimately what this does is maintains this gradient of protons, a high level of protons in the intermembrane space, a lower level of protons in the matrix. So this accumulation of protons in the intermembrane space is like water behind the dam. It's potential energy. And the protons are going to be allowed to flow back into the matrix. And when they do that, that releases some energy, the built-up energy of the potential energy of water behind the dam. So protons can only go back across the membrane if they go through a channel protein called ATP synthase. So ATP synthase opens its pore. It's actually like a turbine. It's pretty cool. And the protons will flow back through, and as they flow back along the gradient, they will spin the turbine, and that provides energy or captures the energy. As the gradient is being released, it captures the energy to phosphorylate ADP into ATP. So here's a little image of ATP synthase, although there's some other, I'm going to link up a video that you can see that shows the turbine much better. But imagine a very high concentration of protons in the intermembrane space. Imagine this pore that allows the proteins to flow back, and it spins, this is the turbine here, it spins the turbine as it flows. The hydrogen ions flow across, spins the turbine, and that produces the energy needed to join ADP plus PI to make ATP. So the ATP is recapturing the energy of the gradient in a sense. And then what you have when you look at everything put together, here's the electron transport chain. Nearby is the ATP synthase. Sometimes ATP synthase is called complex 5, but I'll refer to it as ATP synthase. But remember the NADH and the FADH2, which are produced from glycolysis, pyruvate oxidation, and the citric acid cycle, now come to complex 1 of the electron transport chain, release those high energy electrons. Those electrons travel along the membrane and end up being combined with oxygen to make water. But their energy was taken from them in order to pump the hydrogens into this intermembrane space to create pressure. And then when that pressure is released, when the protons are allowed to diffuse back through the ATP synthase, it turns the turbine of that enzyme to provide the energy to make ATP from ADP and PI. And I have linked up on eCampus and also here in your PowerPoint an animation that kind of shows everything together. And this one comes also from Harvard. It's a really well done animation. Here's ATP synthase over here on the far right. You can see a little better the pore protein and then the little turbine that actually spins. So I would strongly suggest you watch this animation to really get a picture in your head about how this works, because it is fairly complicated. Now, all 4 steps of respiration, so glycolysis, pyruvate oxidation, Krebs cycle, and this last stage, ETC-CHEMIOSMOSIS-OXIDATIVE phosphorylation, altogether theoretically should yield between 36 and 38 ATP per starting glucose. Remember, we started glycolysis with 1 glucose, and we should end up with about 36 to 38 ATP. Actually, what you end up with is a little bit less than that. There's a little bit of loss due to, sometimes the inner membrane is a little bit leaky to the protons, and sometimes the proton gradient is used for something else. But I usually say something in the 30s is a good number. So on the test, if I ask you how many ATP are made from 1 glucose, pick the answer that's in the 30s. So I'm not going to really distinguish between 30, 32, 34, 36, 38. All of those could be correct. depending on the conditions of the cell. But it's something in the 30s per glucose. Now remember, I told you glucose has enough energy equivalent to 100 ATPs. So that means you're only getting something in the 30% efficiency of transfer. We're transferring, remember, we're transferring the energy from the bonds of glucose into the bonds of ATP. And so that transfer is only about 30% efficient. Yeah, it's not great, but apparently it's good enough. Now, anaerobic respiration works exactly the same way as what I've just told you with all the 4 stages and everything. Some organisms don't use oxygen, though, as the final electron acceptor. All right, so sulfur is a pretty common one. Sulfur behaves chemically a lot like oxygen, and it can be the final electron acceptor for certain organisms. Fermentation, though, is fundamentally different. Fermentation is basically glycolysis with a little bit of a modification. So fermentation is not at all efficient in transferring energy from glucose to ATP because you only get, remember, from glycolysis we only got 2 net ATP. So fermentation will take 1 glucose and give you 2 ATP, which is only 2% efficient process. But for some organisms, it's necessary for them to do it that way. In fermentation, the final electron acceptor is some kind of carbon compound. Usually it's either pyruvate or something like that. There's 2 types of fermentation that we usually mention in intro biology. One is lactic acid fermentation. That happens in human muscles, well, other animals as well, but human muscle cells can do lactic acid fermentation if there's no oxygen. So it's their second choice. But if there's no oxygen available, they will do lactic acid fermentation. And yeast will do ethanol fermentation. So this picture is just showing a little, that you're actually looking from like the satellite above the Earth, and you can see a bloom, like a growth. It's a fairly large growth area because remember, you're looking from a satellite. You can see that little green haze represents a huge growth of hydrogen sulfide bacteria. So they're doing anaerobic respiration for their growth process. But anyway, let's talk about fermentation. In fermentation, you basically have glycolysis, so glucose is converted to 2-pyruvate. And then those pyruvate, for lactic acid fermentation, pyruvate is converted to lactic acid or lactate. So you only get the 2 ATP out of this. But your muscles will do this when they run out of oxygen. So if somebody drowns, they can tell if somebody drowns based on the amount of lactic acid in their muscles because they're when they run out of oxygen, their muscles will switch into lactic acid fermentation for a little while. But if somebody is murdered and thrown into a lake, then you wouldn't have that lactic acid. So it's interesting in terms of forensics, they can use some of this to identify cause of death, for example. But another interesting kind of fermentation is ethanol fermentation. And it also uses pyruvate as the final electron acceptor, but it produces, instead of producing lactic acid, the enzymes produce ethanol and carbon dioxide. And so for yeast, it's their way of getting energy out of glucose. And the waste product is the ethanol and the CO2. But for us, it's a product that we like, and it's been very important. Actually, winemaking and beer making was the basis of modern microbiology as a science. So it's kind of interesting. When you take microbiology, you'll learn some of that history. But anyway, so fermentation as a business for humans is very a very big business. For the yeast, it's just a way for them to get energy from glucose into making ATP. All right, but let's go back to the full aerobic respiration. In aerobic respiration, you have glycolysis, pyruvate oxidation, citric acid cycle or Krebs cycle, and then that last stage, which is electron transport, oxidative chemiosmosis, oxidative phosphorylation. The summary equation for all of respiration, aerobic respiration, always starts with glucose, but in reality, your body is able to use glycerol, but glycerol can be converted into another 3-carbon molecule like pyruvate. Some amino acids can be converted into some of the intermediates of glycolysis. And your body can use other sugars like fructose instead of glucose. So at various points in glycolysis, at various points in that 10-step process, different food molecules can enter into the pathway and be oxidized. Fatty acids are typically oxidized starting at pyruvate oxidation, and some of your amino acids will enter into the path at that point, and then other amino acids will enter in at the citric acid cycle. Like some of your amino acids can be converted into that 5-carbon structure called alpha ketoglutarate, for example, which was one of the steps of the citric acid cycle. But anyway, so what we're saying is that your body can oxidize other monomers. One thing that your body never oxidizes for energy, though, are nucleic acids. So carbohydrates, yes. Fatty acids and glycerol, which are the components of fats, yes. Amino acids from proteins, yes. But not nucleic acids. Nucleic acids are not oxidized to get energy. They do have energy, but the body does not break them down. All right, so this is just a picture of the, you do not need to memorize this at all, but these are the names of the different amino acids. I don't know if you know any amino acids, but it's just showing that they can be converted into some of these molecules which are already part of the citric acid cycle. All right, changing gears a little bit. Let's talk about how glucose gets into the cell in the first place, because that's the starting material for our overview equation. Glucose plus 6 oxygens eventually yields 6 CO2 plus 6 waters plus something in the 30 ATP range. So how does that glucose get into the cell in the first place? I think you have a sense that there has to be some kind of channel protein or pore protein, and there is. But the cells don't have that protein on their surface. What's interesting is the glucose transport protein is called GLUT4. GLUT4 normally is stored basically in the cell on a vesicle. All right? And when your glucose, your blood glucose level is raised, let's say you eat a bunch of sugar or something full of carbohydrates, that stimulates your body to release insulin. That's a hormone released from your pancreas. Then insulin goes through your blood and attaches to receptors on your cells called insulin receptors. And when that happens, that sends a signal into the cell, which causes the vesicle that's carrying the GLUT4 receptor to fuse to the plasma membrane. So you can see here on the left, the vesicle holding the GLUT4 receptor, which is the GLUT4 transport protein, I should say, is inside the cell. But on the right, you can see that that vesicle, shown in purple, with the glucose protein, is on the surface. And when it's on the surface, it opens and the glucose moves into the cell just by diffusion. So it's a facilitated diffusion. But only the receptor, the transport protein is only at the surface when insulin is causing that to happen. If there's no insulin in the blood, then the receptor, the transport protein moves back into the cell on the vesicle. So this is a reversible process. But the glucose comes in, and it actually then, once it's inside the cell, remember that glycolysis just takes place in the cytosol. So it can immediately enter that biochemical pathway for glycolysis. If there's too much glucose in the cell, though, the cell will then attach it together to make glycogen. So either the glucose will immediately go into glycolysis, or if there's excessive amounts, the cell will make glycogen. Now, glycolysis can be regulated, meaning turned on or turned off. And these are the 10 steps of glycolysis, you might remember. You don't have to memorize them. But remember that enzymes can be inhibited. And sometimes enzymes are inhibited by the final product of a pathway. We call this feedback inhibition. So you still have to remember feedback inhibition because we're about to apply that concept. The final product of all of cellular respiration is to make ATP. And when there's enough ATP in the cell, the ATP will actually inhibit the enzymes, for example, that I'm circling here. Now, you don't have to memorize the names of the enzymes, but when there's too much ATP, not too much, but when there's enough ATP in the cell, the ATP itself, which is the end product of cellular respiration, will inhibit the enzymes that are involved at the very beginning of cellular respiration, specifically glycolysis. So that's an example of feedback inhibition. For the last slide, I have this picture. This is you really even can't even read it, but this is a poster that would fill up an entire wall. At a university in a biochemistry department, they would have this poster, and it would fill up the whole wall. And it has, you can't read it because the print is too small, but it has every single biochemical pathway of the cell and how they interconnect, and all the molecules and all their names, and all the enzymes and all their names. But what I want to show you is at the very middle of this is the Krebs cycle. And it is really like the central point of all the biochemistry of the cell, for the eukaryotic cell anyway. So you can see it. Let me erase my mark here. You can see it in the center here. So everything kind of converges, or it's almost like one of those turnabouts, where everything in the cell feeds into and spins off of something involved with the TCA cycle or Krebs cycle. So that was just something I wanted to show you. All right, make sure that you watch the accounting, respiration accounting activity videos and fill out that worksheet. And also that little video that I had earlier about the electron transport chain that was put out by Harvard, I think is very helpful.